Okay, guys, let's kind of start with the lab number 4, Protista, and we'll start with the pre-lab questions. How many supergroups are there? Well, in the lecture, we talk about 6 major groups. In the lab, it's talking about the 7 main group. We didn't cover this stromatophylla, but that's okay. So 6 or 7, let's kind of decide on that one. I think 6 would be just fine. So again, if you'll go into that particular later on, if you'll decide to go into a zoology of invertebrates or protistology, you will cover all of those particular groups. But if you know those 6 major ones, you should be okay. Now, describe talk about sexual, asexual reproduction with the bacteria. So, asexual reproduction, the offspring genetically identical to the parents. During the sexual reproduction or sexual reproduction, asexual reproduction is the process when the parent is genetically identical to the offspring, or offspring is genetically identical to the parent cells. Describe sexual reproduction. Now here we start talking about genetically different. The parent would be genetically different from the offspring, or offspring would be genetically different from the parent. So keep that in mind. Are the protists unicellular or multicellular? They can be unicellular predominantly, they could be colonial, and they can be multicellular as well. Different species, very, very diverse group. Are the protists prokaryotic or eukaryotic? They belong to domain eukarya. For some of the students, it's like, well, if it's a single cell, it has to be a prokaryotic, right? No, that's wrong. If it's a single cell, it could be either prokaryotic, then it's a bacteria, or it's a eukaryotic. Then it's most likely a protist, because most of the fungi are multicellular, plants are multicellular, and the animals are multicellular. Now, in this particular case, protists are eukaryotic organisms. They have a nucleus and membrane-bound organelles, endoplasmic reticulum, Golgi apparatus, mitochondria. Some of them have chloroplasts, etc. Do protists reproduce sexually or asexually? Both. They have a sexual cycles and they have asexual cycles. Are the protists heterotrophic or autotrophic? Both. Some of them are autotrophs, some of them heterotrophs, some of them something which is known as a mixotrophs. What does it mean? If the light is available, they are photosynthesizing. Light is not available, like, okay, I'm hungry, I need to eat someone now. How long did life appear on Earth? This was in the last lab. About 4 billion of years ago, give or take few hundred millions. Some scientists saying 3.8, some scientists saying 4, some scientists saying 4.2. I read the article last year, I think, that the scientists discovered some of the rocks, which is about 4.2 billion of years old, and presumably they found the fossils of the cyanobacteria. But yeah, around, to be on the safe, around 4 billions of years old, 4 billions of years of life. We have it here on Earth, while the Earth is about 4.6 billions of years old. How long did the sexual reproduction evolve on Earth? Wait. I skipped one. So number 9. How long did eukaryotic cells appear on Earth? About 1 billion of years ago. So prokaryotes, bacteria, has been on this planet for 4 billions. Eukaryotes for about 1 billion, even though there is some of the indications that some of the groups might be of eukaryotic organisms might be here a little bit older than that, but let's be on the safe side, 1 billion. How long ago did sexual reproduction evolve on Earth? Around probably 3.5 or so billions of years, because remember, bacteria already have a very curious way of sexual reproduction. Name 2 ways humans benefit from the protists. Anytime we are going for human benefits, the number 1 benefit for most of those groups, we eating them. Yes, we do. We eating protists. Like, how are we eating protists? Anybody tried sushi before? The black wrapper around the sushi? That's the algae. That's the dried up algae, right? Algae are protists. What would be another way the humans benefit from the protist? A medicine. Food and medicine. That's almost 90% of all the groups we will be able to answer. Hey, the fungi, food and medicine. Plants, food and medicine. Animals, food and medicine. All right, kind of keep that in mind. All right, so table 1, unicellular protist models. Kind of, let's take a look. This is what you will see in the actual lab. And this is 3 models you need to be able to recognize, name the organism, amoeba, paramecium, euglena, and then to be able to name some of the structures. Which type of the protist use the pseudopodium? Amoeba will be using the pseudopodium to move. Which type of the protist using the flagellum? Euglena. Which type of the protein right here? The paramecium. Which types of the movement will be fastest? Because you have many of them and they're beating all together, propelling the organism forward. Which those types would be the slowest? Amoeba, to grow a false foot, and then another one, and then move. So that's kind of slow. Which of those protists are heterotrophic? And how do you know? Well, if you'll see some sort of green structures, such as chloroplast, that's most likely the autotrophic. Do you see green chloroplasts inside of the amoeba? No, heterotrophic. What about paramecium? No, heterotrophic. So amoeba and parameciums are heterotrophic organisms. Now, euglena. Here's a tiny little bitty thing about the euglena. Euglena is generally, everything's good, generally photosynthetic, but also it's something which is known as a mixotroph, and this is why it's not put in a plant or animal part. It can move actively with the help of flagella like an animal. It can photosynthesize like a plant. Also, if you put euglena in a dark environment for quite some time, for a day or so, it will lose most of its chloroplast and will start actively hunting for the bacteria or smaller protists like an animal does. So see, this is what the protists are. They are not well-defined fungi, well-not-defined animal or plant. Now draw the following protists, and I have a nice pictures of that right here. You can literally print screen it and put it right there. Literally yesterday I had a student sending me the lab. He or she uploaded the Word doc as a lab 4 and then sent the PDF to me. I'm looking at the PDF. Everything is filled out, but there is no pictures whatsoever, no labeling. That's about half of the job. So be careful. You have all of those images. All you need to do is basically move it from one to another. Euglena and paramecium. You need to be able to recognize those ones. Make sure that here we have some of the major structures which you need to be able to recognize as well. I don't think it says label, but in this particular case, make sure that that particular stuff right here in between the organelles is the cytoplasm, cell membrane, the outer part, that's the contractile vacuole. That's the nucleus, a pseudopod. Some of the students asking, what about this? That's most likely the food particles or food vacuoles. That's most likely mitochondria. I would not be asking you about that. But any of whatever is labeled food vacuole, well, contractile vacuole, cytoplasm, nucleus, pseudopodium, make sure to know what they are. Here we have some of the pictures of the preserved amoebas under a lower magnification, and then we're going at the higher magnification. You can clearly see the outline, so cytoplasm, nucleus, pseudopod. Okay, and I think that's pretty much it. And again, I already told you about the different colors. That's a different dyes. This is how the amoeba look like alive, so to speak. You can see those projections are pseudopods and close up on the same amoeba. Now the paramecium, you will see quite a few structures. This is how it closed, this is how it opens up, and you will see two of those kind of star-looking structures. That's a contractile vacuoles because it has a very high speed, the more water moving in. So we have a large nuclei, we have a small nuclei, macronucleus, micronucleus, and those orange yellowish balls are nothing else but the food vacuoles. So, but again, in this particular case, yellow stuff is the, right here is the cytoplasm, the outer membrane, cell membrane, macronucleus, micronucleus, and then we're looking at the two contractile vacuoles. And then, of course, we're looking at the outside layer is the contained bunch of cilia. So here we have oral groove. You don't have to worry about the oral groove, but contractile vacuole, micronucleus, and cilia, make sure you know those ones. And this is how it looks as the preserved specimen. You can clearly see the nuclei, a large one. You can see kind of fuzzy things outside. That's the bunch of cilia and the cytoplasm. And you'll see two white spots or lighter spots. That's most likely contractile vacuole, but I would not be asking them on the live specimens. Oh, this is the live specimens actually. Here you can see the contractile vacuole really nicely. And then euglena, we talk about that. This is the front end, flagellum, of course, the euglena itself. This is the nucleus. Here we're looking at the chloroplast. This is the contractile vacuole, and that particular one known as the red spot or eye spot. Why it's necessary? Once again, sensing the light, so euglena can move toward the light so it can photosynthesize. There is a question quite often, typically you will see on the lab practical, is euglena or identify the organism, you say euglena, and the next question will say, is it autotroph or heterotroph? You can answer, if you answered autotroph, I'll be okay with it. If you'll answer both autotroph and heterotroph, also known as a mixotroph, I'll be okay with it as well. If you'll say that it's a pure heterotroph, I would not be okay with that because it's actually can photosynthesize and it's preferred to photosynthesize rather than the go actively hunt something. So here we're looking at the chloroplast, yes, central vacuole, already talked about that, all of this. And euglenas are quite small, as you can see. This is a 400, this is 100 magnification. Those tiny little biggie specks are euglenas. So if you look at the higher magnification, especially like right here, you can kind of see the green stuff, and then in some cases you can actually see the flagella coming out. But yeah, flagella is usually, this is the live one, so the flagella is constantly beating, so kind of hard to. This one, you can see flagella nicely. You can see that one right there. That's a part of the flagella. So this is the front end of the euglena. Let's see what's the next slide is telling us. So next slide is the unicellular Protista, and in this particular case, we're talking about the parasites. So parasites about the trypanosoma, and trypanosoma is the protist, which in this case it's causing the sleeping sickness. Sleeping sickness, essentially those particular trypanosomes first starting in the blood, then they moving on into cerebral spinal fluid, and eventually in the brain cerebral fluid. As a result, the person who has that over a period of time will become kind of lethargic, does not want to do anything, want to sleep, constantly tired, like half of us right now. So in this particular case, a tap from the, well, basically a blood will show a picture which looks something like that. And you can clearly see those particular trypanosomes, red blood cells, everybody can see that, and those particular things. In this case, you can actually see a part of the undulating membrane. Now, how the person will get it? There is something which is known as a vector. Vector is basically how the person will get it. Like, for example, with the coronavirus right now, what is the vector of coronavirus? How a person, a healthy person, will get the coronavirus? Oh yeah, eating the bat, that's one of the ideas. But no, right now the main vectors for the coronavirus is the people who are sick. In this particular case, the vector for that particular sleeping disease, sleeping sickness, would be a specific type of the fly. It's called tsetse fly. Sometimes refers as tsetse fly, whatever your, well, whatever this is the spelling of that. So in any case, that particular fly is generally parasitizing. It's a blood-sucking fly, which is going for the large herbivores, zebras, antelopes, cows, etc. But occasionally can bite a human being and then infect them with this particular stuff. And I think there will be a question regarding the other possible vectors. So in this particular case, we're looking at the Trypanosoma rhodensis, the creature which has the undulating membrane, flagella or undulating membrane. And in this particular case, you need to draw, take a print screen of that, and then the pinkish stuff would be a red blood cell, and those squiggly looking thingies, that's the trypanosomes. So where do the, in the human body would you find this protist? Well, that's a blood cell, so naturally in the blood. Examine the shape of the protist. Do you think this organism moves by flagella, pseudopod, or cilia? In this particular case, you can clearly see the pseudopod or / undulating membrane. It's an additional part to reinforce the flagellum. A single flagellum with undulating membrane, because once again, the blood is much more viscous than the regular water. Is this protist heterotrophic and autotrophic? And some of the students like, I'm not really sure if it's a heterotrophic and autotrophic. And usually my answer is like, I don't know about you guys, but inside of me is pretty dark. Literally, not the soul or my mental statement, but inside of me, it's dark. There is nothing, no sun shining inside, right? If there is no light, the photosynthetic organism will not be able to survive. That one is definitely a parasite. It's a parasite, meaning it will take, or it will eat the host. Well, I don't have a trypanosome, but human being, for example. It's absorbed the nutrients from the host. So it's definitely a heterotroph. Is this protist multicellular, or unicellular, or multicellular? It's a single cell organism. Lots of them, but still single cell. You can see this is our cell. This is about the same size, but the extended or long, elongated cell. So a single cell organism. What is another possible vector? Well, the main vector for this particular one is a tsetse fly, which is a blood-sucking fly. Whenever it bites, it's injected with the saliva. We just talked about the same thing with the mosquito. So technically speaking, mosquito could be another vector. Well, it doesn't really work that way, but it is possible that the mosquito is another vector. What else? What else sucking blood? Any other things which taking our blood, biting us? We're talking about fleas. we talked about lice before. Technically speaking, ticks can do that or carry some of the diseases. Then the leeches. So all of those can be, or another human, vampires, right? Yeah, vampires can do that too. So in this particular case, that would be another possible vectors. All right, paramecium reproduction. Remember, they have sexual and asexual reproduction, and there is most likely you will see that on the lab practical. So be able to tell sexual and asexual reproduction from each other. So in this particular case, let's take a look at the picture. Is it sexual or asexual reproduction? We're starting with one cell, which splits into two cells. That's a mitosis. That's asexual reproduction. Genetically, genes, the amount of genes, the type of the genes in this cell are exactly the same as in the two of those daughter cells. They are identical. Parent cell, daughter cells, the genes are identical. Asexual reproduction. Okay. Will the offspring be identical to the parent or genetically unique? They are identical. They are clones. So far, so good. Any questions, any problems on that? We started with one. We ended up with two. Asexual reproduction, mitosis, with a cellular division later on. Now, here's a higher magnification slide, same slide essentially, but now we start seeing the one splitting into two. Now, reproduction, sexual reproduction. In order to have a sexual reproduction, they have to exchange the genes with each other. So you will end up with a pair. You can see that here, two parameciums are literally merging with each other, conjugating. And later on, they will exchange the micronuclei. Can we see that? Probably not. But in this case, you can clearly see that two of them are conjugated, third one is not counting, it was just there. So, but those ones, two cells merging in order to exchange genetic information and then separate from each other. So here what we have. Two cells merge, that's in order to exchange the genes. So the resulting cells would be genetically unique from the parent cells. Make sure that you will be able to determine which one. From 1 to 2, that's the asexual reproduction. 2 merging, that's a sexual reproduction. All right. So this is a sexual reproduction. The offspring would be genetically unique. And nucleus, which will be exchanged during the conjugation, would be micronucleus. Remember, it will undergo the meiosis first, creating the varieties, then 2 of them merge, and then they exchange. Well, they will exchange, and then they'll merge, and all that stuff. Any questions, any problems on that? All right, so here we have that particular part. Now, chlorophyta, chloros. Chloros from Greek means green. Phytos means plant. It's not really a plant, it would be green algae. So chlorophyta, green algae. Later on, we'll talk about rhodophyta. Rhodos means red. Phytos means plant, so red algae. So chlorophyta. Let's kind of take a look. This is the colonial known as the volvax. Here we have volvax, so a bunch of them. That's the major or adult colony with the dots inside, which is a smaller, something which known as the daughter colonies. So those daughter colonies continue to grow, and eventually they will rip the parent apart and they will swim away. Okay. That's considered to be asexual reproduction. Do they have sexual cycle? Yes, they do. We're not going there. For the volvox, it's quite complex. So in this case, identify volvox. Is it a red, green algae? And now if it's pointing at the main colony, the pointer for the lab practical. If it's pointing at the main colony, it's the adult colony or mature colony, if you wish. And if it's pointing somewhere in the middle, don't say nucleus. It's not a nucleus. Especially, here we go. Each one of those tiny little bitty dots are separate cells. That's a colony with the daughter colonies inside. We have a colony with a tiny little bitty daughter colony. And on some of them, let's see, somewhere I saw. Oh, here we go. On that one, you see you have an adult colony, you have a daughter colony, and within the daughter colony, you have, I guess it's a granddaughter colony already forming. Quite interesting. So anyway, volvox, adult colony, daughter colonies. That's pretty much all you need to know. The volvox slides reproduce sexually, asexually. Here they reproduce asexually, but they do have a sexual reproduction. We're not going there. Yay. Is it heterotrophic or autotrophic? Well, it's a chlorophyta, green algae. That would be autotrophic, photoautotrophic, technically speaking, but that's autotrophic. They photosynthesize and they green, hence the name. Next one is the spirogyra. Looks like the threads. And this is what those threads look under the microscope. Okay. Why it's a spirogyra? Because you can see that particular spiral thingy going inside. That's actually a chloroplast. Under a high magnification, you can clearly see that. That's a chloroplast. And those tiny little bitty dots on the chloroplast, right here, right there, right there, that's something which is known as the pyrenoids. This is the concentration of the starches as well as some of the minerals. So a vegetative filament, the whole strand. Chloroplast is that particular spiral thingy, and the pyrenoids are those particular dots. Here we go. You can see vegetative, the whole strand is the vegetative filament. The lines right here are, well, is the chloroplast, and those particular tiny little bitty dots are pyrenoids. That's a non-reproducing, therefore it's called vegetative filament. All right, now, spirogyra in conjugation. Occasionally, well, it can reproduce asexually, no problem, but occasionally two filaments will get together and they will conjugate. They'll create a conjugated tube, they will merge the cytoplasm, and then you will end up seeing a male and a female strands. And you will need to be able to identify which one is which. It's very simple if you know what to look for. So here we have two strands conjugating together. You can see that the cell met with another, created a conjugated bridge, so cytoplasm is the same now, and then we have another one, so they merge. Which one do you think we have left as well as the right filament? Which one do you think is a male, which one is a female? Let's say female, is it the left or right? Again, type it in the chat. What do you think? Which one is a male, which one is female? Female. Is it on the right? On the right or on the left? What do you think? Left. Left. Female is on the left. Everybody agree with that particular assessment? Or somebody will say, no, I think it's a right. Left. All right. Okay. So we have only 3 students, 4, 5, right? Okay, well, we have right. I'm not really sure, just being difficult. Everybody's saying left and you're still walking, asking us the same question. So maybe it's right. Okay, let's go back to very basic. What is considered to be a male, what is considered to be a female from the basic biological point of view. We're not talking about self-identification, so anything like that. From the biology standpoint, what is the male, what is the female? Well, first of all, we have, well, whenever the conjugation occurs among any kind of cells, what's happening there? The males, they are the givers. Yes, you're welcome, guys. I am a male, so males are givers of what? Of genetic information. We are donors of the genetic information. Okay, now what the females are, they will be recipients. They are accepting the genetic information, combining it, and then they're taking care of this particular future, whatever the baby it might be. That's really important because on the very next lecture, we'll talk about the fungi, and the fungi don't really have male or females, and we'll talk about that why. But again, remember, the males are typically donors, they're giving out the genetic information, and the females are recipients. So let's take a look on that particular picture. We have one filament. We can see that the cytoplasm, or specifically genetic material, being transferred, but on that part, it's kind of hard to tell where it's transferring. Is it transferring to the right or to the left? If we look at this particular picture, on that particular cells, we can see that that particular cell is pretty much blank, empty. Why? Because the genetic material has been transferred to the right, from the left to the right. And now we're looking at what? Algae baby, known as a zygote. This is where the male and female DNA, that one most likely is the male in this situation. This one is the recipient, so most likely is the female. You can also see the zygote on this side. You can also see zygote on that side. All right, so if you'll see something like that, and it will say which one is the male, which one is the female, or like arrow, is it a male or female? You say, well, that one is the male. That one, in that particular case, the thought process, the logic, why it's a female or why it's a male, I could tell you like, oh, that's a male, that's a female, but why? That's the question. And in this particular case, males will donate its genetic material. So the donation happens right there, transfer. And therefore, we are looking at the zygote. So now sketch the picture. So print screen it, or there is what, snap it, right? Or snip it. You can snip it right here, put it right there, and all you need to do is label male filament. In this case, it will be on the left, female on the right. Conjugation tube is that particular structure between two of those cells, the bridge, and now the zygote, the future baby. See right here, that one. That's a zygote forming, the transfer occurs. That one is already, seems like formed already, and that one is also a zygote, but that one is clearly zygote. Do I have any other pictures? No, I don't. So that's the reason why it's a male, that's one is a female. Please do not snip it, that one. I'm giving you as the reference. So use the one which is unlabeled, put it and label it, please. All right. Otherwise, you're forcing me to stop showing the or giving you the references. All right, so this is how it looks like, bunch of threads, bunch of filaments, tubes. That's a dried up spirogyra. Dried up spirogyra, dried up pretty much samples of the plants known as a herbarium. Herbs, right? Herbs, herb plants. Dried up samples, that's a herbarium of spirogyra. Occasionally there will be like, name this. And the person will say, it's a herbarium. I'm like, well, that's a dried up what? If you'll say spirogyra or green algae, I'll be okay with it. But if you'll just answer herbarium, no, I'm not okay. So let's take a look on that. There were several questions. Is this a form of sexual or asexual reproduction? We have male, female, we have a zygote, we have transfer of genetic information. So that's a definitely sexual reproduction, because the zygote is genetically different from a male and female. There is no such a thing as male or female spirogyra. See right there. Why do you think one we call the male filament or another a female filament? Because of the transfer of genetic information, genetic material. The male is the one which is giving out, donating the genetic material, and the female, the one which is accepting, right? Okay. Now, herbarium species, so specimens. This is dried up specimen of what? Of spirogyra. Do you have to draw it? Well, yeah, you can do a bunch of lines if you want to, or you can again take a picture or print screen, whatever, and insert it there. Not a very hard one. Label, by labeling, we're talking about what is it? Spirogyra. Next one, the green, called sea lettuce. You don't really have to memorize that sea lettuce, that it's a ulva, but in this particular case, yeah, be able to recognize. Most likely the question on lab practical will say, name the type of the algae or protist, and you need to say green algae, or which group of the algae it belongs to. It's a green, it looks green. It's a green algae right there. Do you think it's autotrophic or heterotrophic? They green, why? Because they have a chloroplast, because they are photosynthesizing, so they are autotrophic. Is it multicellular or unicellular, or multicellular? A rule of thumb, which works in 90% of the cases, if you can see it with your naked eye without the use of the microscope, and that particular herbarium is about the size of this. That's a herbarium right there. So it's definitely visible with the naked eye. Visible with the naked eye, most likely you're looking at a multicellular structure, so a multicellular organism. Yes, there is some exceptions from that particular rule, like anybody seen the egg before, the chicken egg, that's a single cell, but that's the exception. Rule of thumb, if you see it with your naked eye without the use of the microscopes, then it's a multicellular organism. All right, next group, table 5, known as a phytophyta, also known as a brown algae. You can see them kind of brownish color, see right there. So this is a herbarium of the microcysts, and you will need to draw and label few major things. Also know the function. All right, this is the overall dried up plant, and dried up plant will attach itself to the bottom of the ocean or sea. It's usually lives in a shallow area, so to the substrate with something which is known as a hold fast. Those root-like structures attaching its it's not roots. That's why they have it hold fast right here. Now, on top, we'll see a blade-looking structures, a leafy-looking structures. That's known as the blades. The primary function of the holdfast is attached to the substrate. The primary function of the blades is photosynthesis. So far, so good. Now, many of those particular things will have something which is known as a float, also known as an air bladder. Why it's needed? It attaches to the bottom. If there is no air bladder, it will sink to the bottom, where you don't have much of the light. Closer to the surface, more light you will have. As a result, the float will bring the blades toward the surface. More photosynthesis, more food, bigger it can grow. And now to connect this particular combo of the blades and the bladder or air bladder with the holdfast is the stem looking structures. It's not a stem yet. That's why there is a specific term known as the stipe. So what is the function of the blade? Photosynthesis. What is the function of the holdfast? To attach to the substrate. What is the function of float? To bring the blades closer to the surface for more photosynthesis. More photosynthesis means more food. So far, so good. Any questions, any problems on any of those structures? Presumably not. Moving on to the next one. No. Okay, all right. Thank you. Here we're looking at the very common algae. We have it here in the Galveston Bay. It's lacking one of the major things. And it does not have the holdfast. It's just free floater. It does not attach itself. It has everything else, but no holdfast. So let's kind of take a look. What do we have? Well, those ones are smaller leafy structures are the blades. They're much smaller. Those things which are connecting blades to each other, this is the stipes. Those small dots all over the thing are the. As a result, that particular algae literally floats on top of the ocean, periodically creating something which is known as drifts. Those drifts can come out on the shore, and then the beach, for example, 2018, middle of the summer, again in Cancun area, I was spending some time there. And there was a tons of drifts coming of sargassum coming out on the shores. So in some areas, instead of a nice, really nice sandy beach, you would have a layer, literally 15 to 20 inches of dead, drying out, stinking up algae. In some of the areas, of course, especially near the major hotels, they were cleaning up literally every day. But in some of the areas, you can see like literally like a mile of shore is completely covered with those particular drifts. And of course, during the early sailing times, whenever we had the ships with the sails, getting on one of those particular drifts was a really, really bad thing because you slow down drastically. So yeah. Which structure blade floats type of holdfast sargassum do not have, and that would be the holdfast. It's not connecting to the bottom, to the substrate. What does it tell you about this algae? That it's a free floater. A few examples of commercially available brown algae has been provided. So again, we have the labeled picture, and some of them here we have roasted seaweed snack, brown algae. Remember agar, agaros, we're looking, whenever we're looking at the bacterial plates, the bacterial plates had the agaros, and the agaros are that particular gel-like material. We're using it for, well, besides the growing bacteria, we're also using it as the gelatin, especially if the people are going for totally vegan diet. They cannot have gelatin, jello, because gelatin is made out of or extracted from the bones as well as the tendons and ligaments of the animals. Vegan cannot have, so as an alternative, a very healthy alternative is agar-agar, so thickening agent. And then the chlorella powder, which is a green algae, you can buy it as the superfood nowadays. As a matter of fact, chlorella is one of those single-cell organisms, plants, which we are thinking might be the primary food source for the astronauts to go to the different planets, because, well, they have sun, so they can reproduce, and then you're drying up and eating that stuff. So yeah. All right, next group, Rhodophyta, red algae. You can kind of like, well, I'm not really sure if it's comparatively to the brown algae, right? So reddish tint, that would be the red algae, porifera. And in this particular case, it looks very similar to this guy right here, right? Look, green algae, red algae. Well, first of all, they are related. Second of all, they do the same function in the environment. As a result, they start looking alike. And they are autotrophic. They still algae. They are, even though they're using, besides the chloroplast, they're using some other pigments or other plastids. Okay, and the red algae, we are consuming it. Yeah, food. Now, Chrysophyta, golden brown algae, golden algae. In this particular case, we're looking at the diatoms. You see those shells made out of silica, right? SiO2, silica dioxide, and glass, essentially, glassy ones. And they're usually made out of two parts. So diatoms, because they are made out of silica, whenever they're dying off, they settle down on the bottom, creating something which is known as a diatomaceous earth. And diatomaceous earth is used for quite a few things. One of them as a medicine, a natural medicine to get rid of some of the worms. If the person has a worm, the swallowing a spoonful of diatomaceous earth will essentially kill those particular worms. Why? Because, well, again, you are swallowing microscopic shards of glass. And whenever it goes through your digestive system, your cells will not be affected, but the cells of those particular, well, with the patient, worms might get killed. A friend of mine, he's all natural, and he's using diatomaceous earth to kill the fire ants. He sprinkled diatomaceous earth on the fire ant hill, kind of poking around, so the ants will start attacking the diatomaceous earth, swallowing some of those particular glass shards, and it will rip them or cut them from inside. And I'm like, you're horrible. He's like, what? Why? It's very, very ecologically friendly. I'm like, you're horrible to the ants. In my case, you're taking some sort of insecticide, spray it within an hour, those particular ants dead. That's it. It kills them. It's a toxin. In your particular case, the ants will continue to live for a day or two, but they will be literally having the intestines ripped to shreds. Anyway, so here we have a diatomaceous earth. Oh, and the diatomaceous earth also used for fine polishing, because again, minute shards of silica there. Intricate, made out of silica, diatoms. Make sure you will be able to recognize those for the lab practical perspective. So let's see, what's our next slide is showing. That's it. Oh yeah, that's the last one. So do they have a symmetry? Most of the time they will have something which is known as a radial symmetry. What makes up the shell of the diatoms? Silica, SiO2, silica, or you can say glass. I will accept any of those answers if it will be on the lab practical. What method paramecium used for sexual reproduction? Let's kind of settle that one down. Conjugation would be the proper name for that. I think in somewhere it says, no, for sexual it says the conjugation. One of the advantage of sexual reproduction, genetic variance. Describe the advantage of asexual reproduction. Very fast, don't have to worry about finding a partner. What is method Spyrogyra used for sexual reproduction? Conjugation. Again, merging together. Describe one commercial use of algae, food, medicine. What organism has a holdfast? What its purpose? Many of the algae do, brown algae, purpose to attach to the substrate. Which organisms have male and female filaments? Spyrogyra. Which filament will contain zygote? Remember, males are donors. They will give away whatever they have. The females will accept and then they're taking care of the zygote. So female filament will contain zygote. What do we call asexually produced offspring in adult volvox colony? It will be called the daughter colony. What three structures do various unicellular protists use to move around? Flagellum, pseudopodium, cilia. Fill out the table below. Amoeba, unicellular. Motility, pseudopods. Autotrophic or heterotrophic? Heterotrophic. Parasitic or free living? Free living. Paramecium, unicellular. Motility, cilia, heterotrophic, free living. Euglena, unicellular. Motility, flagellum, or flagellum. Autotrophic / mixotrophic. Let me type it in for everybody. Mixotrophic. Either or. It's it can switch. So it's mixing two things together. So autotrophic / mixotrophic, parasitic or free living. Since it's a autotrophic, it got to be free living. Now trypanosoma, still unicellular. Motility, flagellum / undulating membrane. Undulating membrane, heterotrophic, and the parasitic one. Now, next table of the algae. Common name, see right here, chlorophyta. Chlorophyta means green algae. So green algae, green algae, green algae. Phytophyta, brown algae, brown algae, brown algae. Rhodophyta, red algae. Chrysophyta, golden algae. Spyrogyra, multicellular. Volvox, colonial. Ulva, the ciliatus, multicellular. 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 Unicellular. So far, so good. Distinct characteristics. That's up to you guys. Write something down which will help you whenever you're studying for the lab practical to tell Spyrogyra against the volvox. You can say round colonies with the daughter colonies inside. Spyrogyra, long filaments, thread-like filaments. Ulva, a green large, I had a student last semester. He said, looks like a crumbled towel paper, reddish color. Looks like a crumbled paper towel, reddish color, greenish color. Anything which helps you to identify those in the lab practical, because most likely you will see pretty much every single group of them. All right. And your favorite reflection questions. So once again, complete sentences, at least a paragraph. A single sentence is not a paragraph. I observed evolution. Good, not a paragraph. So give me a little bit more. I'm reading those things. All right. And that's pretty much it. We done for today.